Hello everybody, and welcome back to BioSC 140, Human Physiology. This video is going to cover Laboratory 15 and 16, the experimental procedures. So this is one of my favorite labs the whole semester, actually. It's the sensory system laboratory. And I love this lab because we kind of just trick our senses. We push our senses to the limit. We make our senses feel, experience things that don't make logical sense always. It makes our senses work. We experience how our senses are limited, and I like it. It's fun. So there's lots of little experiments within this experiment. In FOX 3.4, we're examining cutaneous senses. So we're going to map skin receptors. We're going to do 2-point threshold. We're going to do receptor adaptation and referred pain. In FOX 3.5, we're going to do eyes and vision. So acuity, astigmatism, and accommodation. Our first part is mapping touch receptors. So the purpose of this is to see where our skin receptors are and how they're distributed. So procedure, we're going to make a 4x4 grid. So in class, we'll have students put 16 dots, a 4x4 grid with marker on their forearm. Usually the boxes are about 2 inches to 3 inches on each side, and the dots are evenly spaced apart. Once we have the dots drawn on each other's arms, we're going to have the student who's the experimentee, the one who's getting experimented on, look away. They're going to rest their arm on the lab table, and they're going to look away from their arm. Their job is to say something when they feel something. Their lab partners are going to take a room temperature probe, and they're going to tap each one of those dots lightly. If the person says that they can feel it, you're going to record a T for touch in that location. If the person cannot feel that light touch of the probe at that location, you'll leave that spot blank on the data. We are then going to repeat that with a probe that's been sitting in warm water. Now make sure when you do this, you touch really, really lightly so that we activate heat receptors, not touch receptors. So we're going to do this again, and same procedure where the student looks away, and wherever they feel it, the warmth, we're going to put a W. So a W for warm. And then we're going to repeat again, just this time the probe is going to sit in ice water before we do it. So it's going to test for cold receptors. Most of these experiments within this one experiment, you could actually do at home. You could do this with a fork. Be careful not to burn yourself, be careful not to stab yourself, but you could do this at home and experience it. So what do you think is going to happen? Do you think it's going to have the same sensation everywhere? What do you think is going to happen? All right, next up we have the 2-point discrimination threshold. This one you can actually do with a paper clip at home. Be careful, not to stab yourself again, be safe. Purpose of this is density of touch receptors varies throughout the body. What is the density in different body parts? So in this experiment, we're going to use something called an anestisometer. It's really hard for me to pronounce that word. Is it written down? It's in the notes. Anestisometer, it's really hard to say, but it looks like this. And what this has is it has a ruler, and it has these 2 prongs that come out. And what we're going to do is we're going to start out by having the student place their hand on the lab bench and looking away, and have them look away. The lab partners are then going to bring the two arms of the anesthesiometer together, and they're going to tap the fingertip. And the person who's looking away is going to say they feel one prong or they feel two prongs. After the first one, the lab partners are going to move the prongs 1 millimeter apart and repeat. Then 2 millimeters apart and repeat. Then 3, then 4, and so on until the person feels two distinct prongs. We're going to repeat this with the fingertip, with the person's palm, the back of their hand, and the back of their neck. And we're going to see how far apart do 2 points need to be before the person feels two distinct points. Do you think there's going to be a difference? Which one do you think is the most sensitive? Which body part do you think has the highest density of touch receptors? Adaptation of temperature receptors. Purpose. Sense receptors will stop responding to constant stimuli. They will adapt. How will adaptation influence sense of temperatures? So with this one, what we're going to do is we're going to put one hand in ice water and one hand in warm water. So your right hand is going to be in ice water, your left hand is going to be in warm water. If you do this at home, don't burn yourself and don't freeze yourself. For 1 minute. Then the student is going to remove both hands from the water and place it into a tub of water that we know is room temperature. And they're going to write down what they feel. What do you think the hand, so the hand that's been in warm water for 1 minute, when it gets put into room temperature water, what is that room temperature water going to feel like? The hand that's been in ice water, when it gets put in room temperature water, what is it going to feel like? Hypothesize. Referred pain. So purpose. Pain felt not arising from origin. Pain felt somewhere other than where the tissue insult is. So with this one, what we're going to do is we're going to strike the ulnar nerve or the funny bone really lightly, and we're going to record where the person feels it. So to do this, what you want to do is take your elbow, put your thumb or one finger on the medial epicondyle, put one finger or thumb on your olecranon process. Right in the middle, you're going to feel a groove. That is where your ulnar nerve is. Then hyperflex, flex your elbow as much as you can. Tap it with your finger. A lot of times you can get a response just by tapping it with your finger. Tap, tap, tap. Start lightly and get harder. And record what you feel. You can do this at home while watching this video. If you don't get a response with just tapping it with your finger, you can don't use like a nail hammer. You could use the back of a spoon. You could use a heavy pencil. You could use a cell phone, you could use a cell phone. Start light, work your way up. Know that this could hurt. If you've hurt your funny bone, you know this can hurt. You can always just take my word for this one, not actually experience it. Take your time, do it gently. What did you feel after you irritated that ulnar nerve? Where did you feel it? Next up, we're moving into the eyes and vision. So Fox 3.5. Acuity is your ability to resolve objects, your ability to have a clear image of something. So Snellen eye chart. For the Snellen eye chart, you stand 20 feet away. So this is the Snellen eye chart. It's kind of that standard vision chart that most people have seen in the movies. The way you do it is you have the patient stand 20 feet away. You then ask them to cover one eye, and then you ask them what number corresponds with the lowest line that you can read comfortably. Let's say they say 6. You say, okay, can you please read line 6 from right to left? P, Z, C, F, D, E. They get it. If they get it, perfect. Okay, can you please read line 7 left to right? F, E, L, and so on. If they make one mistake and the letter kind of looks like that letter, you say, okay, we'll give them that. Let's move on. Once they start getting 2, 3, or more mistakes, that is the lowest line of this. After that, after you found out what line is the lowest one they can read, ask them what color this line is. And then ask them what color this line is. After you do that, you switch eyes and have them repeat with their other eye. If they read line 6 right to left with their right eye, have them read left to right with their left eye. You know, kind of keep them tricked, you know, so they don't remember the don't let them memorize the order. And then record your results. Perform this with and without glasses if possible. So acuity is your ability to resolve objects. We do 2 experiments regarding acuity. Snellen eye chart is the first experiment that examines acuity. The next one is the 2-line discrimination test. So for this one, we put 2 lines, 2 parallel lines on a white piece of paper, and we have them 1 millimeter apart. We then stand close and we put it on the wall at about eye height. We then stand, so we got the 2 lines 1 millimeter apart on the wall. We stand a few feet back away from it and we cover one of our eyes. We should be able to see that there are 2 distinct lines. You then walk backwards until those 2 lines merge together, until those 2 lines become one. At that point, you stop. You find out what that distance is in millimeters and what that distance is away from the 2 lines. So we have tape measures in lab normally. And so you do the tape measure from the 2 lines back to that point where it becomes one line. And you record how far that is. Do everything without glasses. Do everything left and right eye. Astigmatism. So astigmatism has to do with light not passing through your eye evenly in all areas, an imperfection in your field of view. For this one, we have this chart right over here, an astigmatism chart, and all these lines are the same distance. What we're going to do is we're going to stand 20 feet away. We're going to remove glasses and cover one eye. And we're going to stare right at the middle of this chart. If some of the spokes appear dark or blurred or light, if you close one eye, cover one eye, and you stare at the middle of this chart from 20 feet away, and there's variations amongst these lines do not look equal, then you probably have astigmatism. Don't diagnose yourself in this class, talk to your doctor. Do it for both eyes and then do it with glasses on. For accommodation is your ability to focus on objects at different distances. So in lab, I'm going to explain how we do it in lab, and then I'm going to talk about an easier way that you can do it at home. So remove your glasses, cover one eye, see the instructions for Fox for the measurements, and you're going to measure the near point in centimeters, determine your visual age. So basically, for accommodation is your ability to focus on things at different distances. So you know how like when you take a photo, everybody's taking photos on cell phones pretty much. You know how like if somebody's close, you kind of like tap on their face and it focuses on their face, or you tap on the background and it changes the lens so it focuses on the background. The lens in your camera has to change its shape to see things at different distances, just like how the lens in your eye has to change its shape to see things at different distances. What we want to see is how close can something be to your eye and have it be in focus, have it not be blurry. So what you want to do is cover one eye and hold up a pencil. Hold up a pencil. Stare at the top of the pencil at the eraser and slowly bring it towards your eye. Slowly bring it towards your eye. At what point does that pencil become blurry? So figure out exactly how far away that pencil is. So find the point where the pencil becomes blurry. Hold your pencil right there. Measure that distance. Measure that distance. So here I am. Cover one eye. Very close. Measure that distance from your eye to the pencil tip. And then do it for the other side. And then compare it to expected results. Compare it to expected results for people of your age. So these are the labs. Those are the experimental procedures. I'll see you in the next video with the discussion.